Okay, so we've talked about the plasma membrane, we've talked about the nucleus, and the third structure inside cells that I want to talk about a little bit is the cytoskeleton. This is one that chances are in your intro course you didn't spend a whole lot of time on other than just define it. And that's probably unfortunate because cytoskeleton is actually a pretty active and dynamic part of the cell and is involved in a lot of different things. So let's just think about it in general terms first, and then what we'll do is we'll, over the next few lectures, go through some of the specific structures and some of the roles that this is going to play in development. And of course, one of the main connections here is that the next two structures, this cytoskeleton on the inside and the fourth one that we'll talk about is called the extracellular matrix on the outside. Remember that those two structural components are going to be one of the main mechanisms by which cells tie together into larger tissues and structures. And based on the overall structure of the cytoskeleton and the overall structure of the extracellular matrix, cells will specify into the kinds of cells that they're supposed to be. Of course, another way to think about that is based on who they're supposed to be, they're going to build a cytoskeleton and an extracellular matrix to match. So the relationship here is very much one that is going to help us with this question of how cells know what they're supposed to be and how do they become what they should become inside the embryo. All right, so for this lecture, let's just talk a little bit about what the cytoskeleton is. You guys will probably remember it as a framework of proteins inside the cell, crisscrossing throughout. Of course, the organelles are in there too, so this would be basically in and around those organelles, and it would be a component of the cytoplasm, and a lot of different things going on. So common functions of cytoskeleton and things that this would be doing, it doesn't matter what kind of cell you are, is support. So obviously for as a support structure, it will give the cell itself some structure to it. And by that I mean cells, if you think about cells, they're not just like squishy water balloons, okay? They are, well, a good way to think about it would be if you were to take a balloon and fill it with sand first, maybe put some marbles and other things for organelles, but if you fill it with components and then put water in it and then tie it off, that would be more what a cell is like. So somewhat firm, somewhat not changing in shape very easily, and the cytoskeleton is going to be a really important piece to that characteristic of cells. So the support is largely there to keep cells from being damaged, from being torn apart, from being crushed, and all are going to help that cell maintain its shape and be able to do the things that it's supposed to do in maintaining life. The internal scaffold also helps organize. So in many ways, it's almost like a system of storage compartments inside. I hate to use that word compartments because that sounds more like the organelles, but this is a way in which we can move things to a certain area and then put them there and leave them there. And so that one somewhat is related to the third one here of movement. If we move things around the cell, so let's say we need more mitochondria on one side versus the other, or it's not so much for us, but like in plant cells, we might move chloroplast to the side the sun's shining on. The movement of organelles and other components like ribosomes and other large enzyme complexes, those are going to essentially move along these cytoskeletal elements to get to where they belong, and then those elements, those cytoskeleton fibers, will actually keep them in those locations. So just like we said, the nucleus is not all random and stuff just floating around inside. In a cell, things are not just floating around randomly either. Everything has its position, and the cell can change its positions, or the positions of things, based on the needs it has. So those are the three main functions that cytoskeleton play, and some of the special functions, things that you might find in some cells but not most, would be locomotion. So the motion by flagella, cilia, and pseudopodia are very much related to changes in cytoskeletal structures. So you guys are probably familiar with the flagella and cilia, those things that sort of whip and beat inside of the, or outside the cell. For eukaryotes, the flagella and cilia are actually organelles inside the cell. So there's a membrane that goes all the way around it, and the structure inside that helps it move is made out of the same components as the cytoskeleton is. So we'll see flagella for sure with sperm, and we'll see cilia show up in quite a few places as well. Pseudopodia, not really going to be something we have to worry about too much, but obviously cells migrating inside the body are going to sometimes use pseudopodia to get where they need to go. Of course, in our bodies right now, there are lots of different kinds of white blood cells that use pseudopodia to move around our bodies as part of our immune system at the moment. Cytoskeleton is also used in contraction. So if you've had anatomy, you've studied how those actin and myosin fibers in a muscle cell are going to allow those muscle cells to contract. Well, actin and myosin, well, the myosin is a motor protein, the actin would be a cytoskeleton fiber. So it's a somewhat modified version of a cytoskeleton in muscle cells that allows that to happen. I've already mentioned the connection between the cytoskeleton and the spindle apparatus for mitosis and meiosis, so we'll talk about that a lot. And then the cytoskeleton is also involved in dividing the cell, what we call the cleavage furrow, as the mother cell basically pinches into the two daughter cells at the end of cell division. As a structure, and we'll dig into these a little bit deeper, as we go through them, but the cytoskeleton itself is going to have three different kinds of fibers in it. Each one has a very specific role. Each one of them has a very specific structure to it, and they are all tied together in one large entity called the cytoskeleton. The cytoskeleton is very active and maintained. So you remember we talked about the modular structures and how having subunits and being able to replace things. Cytoskeleton is a great example of that. And the three fibers that we have are microtubules. Those actually stay fairly the same throughout the different kinds of cells. Microfilaments stay somewhat the same, although muscle cells would be a good exception to that. And then the intermediate filaments are ones that tend to be fairly specific based on the kind of cell you are. So we'll talk about those in a little bit too. All of these are connected to each other by a protein called plectin. So plectin is essentially the protein that ties the cytoskeleton into one large organelle or unit inside of the cell. So you can see the microtubules. These would be the red. The orange would be the intermediate filaments. And what's not really pictured would be these actin filaments. You can't really see those here, and you'll understand why as we go through each one of these in turn. So basically what you're looking at in this picture would be a snapshot of what it looks like deep inside the cell, and these guys, the yellow actin filament microfilaments, are located around the surface underneath the plasma membrane. So they probably would not be in this picture here, but you can definitely see those little blue molecules that have that are the plectin connecting the dots here with regards to the intermediate and the microtubules, and the actin filaments would also be in there as well. Okay, so what we'll do is we'll work our way through microtubules, intermediate filaments, and microfilaments, and look at each one in turn, because they do have somewhat specialized functions and structures that make them important. And these structures and functions sometimes are pretty specific to development. So we'll do microtubules next time.